track record of carrying out the fundamental science needed to use phages in applied settings, evidenced by our work on human-associated Clostridium difficile and Salmonella associated with pigs. Much of our work uses genomics and structural approaches to identify key traits associated with phage efficacy to clear infection. Our work also incorporates studying phage bacterial interaction in physiologically relevant models and in animal trials. Our recent publication led to 2 recent British Research Council awards to design phage products for use in livestock, and she also had projects funded directly by the industry and biotics funded C. difficile phage therapeutic programs and other with close industry collaborations. She has also developed and regularly run a course that teaches phage biology to African academics as part of the Gates-funded Phage for Global Health Yale-Leicester collaboration. All of our applied work is rooted in fundamental biology, and early phage research was focused on understanding how ocean bacteriophages controlled their marine bacterial hosts. Our work has paved way for a new research field, which is still very much active, that of determining complex ways that phage interacts with their host. So join me in welcoming Professor Clokie to address our session for this webinar titled The Role of Early Career Research Engagement in Phage Research. So Professor Clokie, you have the floor to commence your presentation. It's very nice to have you with us, and we are very enthusiastic to hear about your presentation this afternoon.
Okay, well, thank you very much, Ibadan, for inviting me to this session. I prepared some slides on the topics that you would ask me to speak about, but I'm very happy for anybody to stop me whenever they want. So it doesn't have to be a formal presentation and then questions at the end. Just feel free to jump in whenever you have a question. So can you see the slides fully now in this mode?
Yes, we can clearly see the slides.
Okay, great. So yes, what you asked me to talk about was a very important topic, which is what is the role of early career research engagement in phage research? So of course, this has to be a little bit of a case study. It's my own personal experiences that I've tried to put together into a sort of linear format. So first of all, I shall talk about my own timeline, sort of what I was doing in my sort of early career research phase, and some things that I think helped me. I'm not sure how many of these things I sort of consciously chose at the time, but it's some things I was aware of and some things I've become aware of. And I'll talk about the importance of engaging in scientific and slightly more general communities as well. So that will be the overall structure of my presentation to you today. So I thought, just so you know, becoming a professor doesn't take, it's not an instant journey. You can see this was my own career path. So I started my first degree in 1996, and then I did a master's degree here in 97. I then did my PhD from 97 until 2001, when I got my first postdoctoral job. And these are a little bit representing the pictures on the top, the subjects I worked on. So even though I'm very interested in phages now, I've actually only worked on phages for about 20 years, probably the same age as many other people in this audience, I'm sure. So I started off working on plant viruses, did a lot of work on plants, and then I moved to study both marine bacteria and phages, and that is more or less where I stayed. So I've been working on phages of many different organisms. This is to represent different bacterial pathogens. This is phages that are sold in Georgia to target many things. So you can see some different phages along the bottom that I've worked on in these different stages. So I became a visiting scholar in 2006. So I was a postdoctoral researcher for about 6 years at the University of Warwick. And then I moved to become a lecturer at the end of 2006 in Leicester. And then the UK system is you become, so essentially a lecturer is like assistant professor, and Reed is like associate professor, and I was been a full professor now since 2015. And I've just added this actually to my slide because very excitingly, as of next year, I shall now be the director of the first UK Bacteriophage Centre. So you can see the time frames are really quite long from obtaining my PhD to start there even until now. It's been about 20 years. So I'll focus in on my early career research, which I'm kind of defining as my postdoctoral and early lectureship years, because I think hopefully, and PhD years to some extent as well. So hopefully the talk will be of interest to people in that age range. And I think it's important to say that during that time, very often when you're establishing your career, you can also be personally, it's very commonly also the time when you're having perhaps your family. And that was certainly the case for me. So you can see here, so if you remember, I would say that I got my job in 2006, but actually I got married here in 2002. And then I had my children a couple of years, and then 3 years after my lectureship. And I think one of the reasons why I was able to make this dynamic work of being having a family and working as a scientist is that very often I involved my students. This was my first ever PhD student with my first son, having a little cuddle during his PhD viva or just after. And sometimes, because when we talk about early research career engagement, it's important to go to things like conferences, but it can be a little bit hard if you have young children. So very often my family and my friends would come with me to meetings to allow me to attend, so I could really engage. And quite often I would just invite collaborators to my home. So here we have a lovely Georgian scientist, Nina Chanishvili. This is our lab family on a lab trip. Some postdocs. We were all on a big trip to China at this point with our families. And this was Betty Cutter, who some of you may know is a lovely phage biologist from America with on a trip there. So sometimes you have to a little bit blur your family and professional relationship to make it work. Right. This I think is really, really key for early career researchers, is that you can actually choose your own mentors. Okay, so you don't just suddenly become a good and successful scientist. You need guidance along the way. And it's really important to choose people that you like, whose work you like, whose personalities you like, to a little bit help you, because it's often not easy, some of the things and the decisions and knowing what to do. So I've definitely done this. And Betty Cutter has been an amazing mentor to me throughout my scientific career since I discovered phages. And these are my three people who were, and still some of them still are on my team, who I've also now tried to mentor. Some of you may know Janet Nally, who's a wonderful researcher who now has her own position in the north of Scotland. These are people. This was my postdoctoral supervisor, who became a great friend and mentor, as did my head of department when I moved to Leicester. This is my favourite lecturer as an undergraduate, who I've always asked for advice along the way. And this is a Canadian phage scientist who I just really liked and loved to chat with him. So I think in terms of early career engagement, you can be quite proactive about who you engage with, just, through people whose papers you like, just chat with them and see what they have to say. So within your early career research progression, you'll sort of progress from being a student. So you'll have things like you'll get your grants, your papers, you'll start to write books, you'll supervise students, you'll have invitations hopefully to go to places, you'll get large grants or commercial funding, patents. So there's lots of these things that you really, that are a big part of the job that will happen along the way. And I think in a way, the more you engage with lots of people, the more you can proactively make these things happen. And there are also these sort of defining moments within your research career where it's important to realize what direction you're headed from, what topics you like. So I realized in about 2001 that I did want to stay working on phages. And then within phages, I spent 6 years working on a particular type of phage before realizing actually I was interested in working in other phage systems. So in terms of engaging within an early research career context, I think it's super important to find a niche for yourself. And certainly as well, when you start working in a department, although you may decide you want to work in a particular area, if you have many colleagues who have some pet projects where you could play a role, I think it's really important to do that, because I used to think that I could only work on these marine phages, but I realized after some years that actually I found all phages really interesting. So I could actually work on phages where other people around me had the expertise in the bacteria, and this allowed me to engage more and progress my ideas further. So I think it's really important to be selective and open to collaborations. So when we talk about engagement, what kind of engagements do we mean? Well, I did things, and I would recommend doing some of these things, like just be very visible. So if you go to conferences, make sure you give talks. You can offer to share sessions. You can even offer to do things as well. For example, people always need people, journals always need people to review scientific meetings, so that's a good way of being seen. So it's really important, I think, to be both quite proactive and quite resilient, and then follow through. And I think sometimes you have to be quite creative with the way you engage. So are there any questions for that from that first part of the presentation, or should I move on to the next part?
Yes, hello. Thank you.
Hello.
It's nice to see you again.
Nice to see you too.
Yes.
Oh, hello.
Hi. I wanted to ask you the last point of your previous slide, where you said be something about chaos. Let me see if I got it.
Oh, yeah.
I just wanted some explanation about that, please.
Yes, I was just meaning that sometimes when you're trying to establish yourself as a researcher, it can seem that there are quite a lot of things happening at once. It can seem a little bit chaotic, but it just sort of sometimes you just have to that you can see there are various things happening, but then you can. I think it's important to not get too phased by that, but to choose within that slightly uncertain framework which things you really want to follow and pursue. So that's sort of what I meant, and also the fact that sometimes I've seen quite often young researchers try to have a perfect family life and a perfect work life, and I think it doesn't really work like that. It can often be a little bit more chaotic. Okay. So it's just a question of not minding that so much. You can't really always have everything at once. So you perhaps have 2 or 3 projects, but one of them will be the main focus of that.
Okay. Thank you.
Okay, that's okay. Thank you for the question. Okay, so I'm going to move on now to give some tips about that relates to going to conferences and workshops. And I think this is really key. So I'm talking to you actually now from a conference in Ireland that I've been attending. And it's the focus of the conference has been about mucus and the way microbes interact with a mucus. And actually, it's been a lovely conference because many of the people here are PhD students, young students who are just starting their scientific career. So it's been, for me, very enjoyable to chat with them. But sometimes I notice at meetings you will have, for example, the young students will just talk to the young students, and then the old professors will just speak to each other. So I think it's really important not to do that. So when you go to conferences, listen to all the talks and just go and talk to the speakers, ask questions afterwards. But at all of the opportunities like at coffee and meals, just try not to be shy. I think I'm quite lucky here because I'm not particularly shy. So if you are not shy, then you have an advantage. And if you are a little bit shy, you've just got to try to be strict with yourself and because people are generally people love to chat about their work. The reason we do this work is because we find it enjoyable. When young researchers come and ask us questions, it's always incredibly enjoyable to hear their reflections on our work. And I think it's also important after conferences when you've met people to sort of follow through. So I always try to take notes during the conference and then reflect on those notes and work out who I might want to talk to so I don't just miss the opportunity. So I've met a couple of really nice people at this conference that I'd like to follow up and see how that bits of their work will help me take some of my ideas forward. So that's I think really important, because conferences are where we go from being just individuals and within an institution to actually feeling like we're part of a much bigger scientific community. So here are some pictures here of a couple of conferences. I went to these are just a little bit before lockdown. The top one was a conference in Malaysia, or workshop. The workshops also are really good to go to because they can be smaller and more focused. This was quite a big Phage conference in China, the one at the bottom. Again, I was presenting my data. This was actually harder because a lot of it was in Chinese. So made me realize how, because generally I'm lucky because obviously English is spoken in most places, but this was not always the case here. So that's some sort of tips on thinking about how we attend conferences, because it's quite easy in a way to just go to a conference and not extract the most from it. You can really use that conference as a excuse to network, you know, find future employment opportunities. And then in terms of how do you actually find a resource to go to workshops and conferences? So one way is to make sure you join professional societies. So for example, the UK Microbiology Society, who's funding this conference I'm currently at, they're really, really good for helping young researchers. And then within, depending on what you're interested in, perhaps the Health Infection Society, the American Society for Microbiology, and basically there are many professional societies out there that if you join, they will give you funding to then help you travel to conferences. And then as you sort of progress through and you get other opportunities, I think there are things like, for example, assessing PhD vivas, and hosting other students in your lab. So if somebody from another university who's another student even wants to learn something you're doing, it's really nice to have them over and you can start to build up your networks at an early stage. And then finally, I put here, it can also be quite advantageous to join teaching efforts. So perhaps if you've got older colleagues who are teaching younger students, you could make yourself a bit more visible within your own institution by helping to do some of those teaching activities. Okay, so that's my sort of focused science, sort of parts of the talk that are focused on specifically on things that you can do to improve your engagement in the scientific paths. So are there any, would anybody like to pick up on any of those aspects before I talk about the advantages of communicating to a wider audience? Can you see? Hopefully you saw that the essentially to summarize that part, I think going to meetings and making yourself very visible at those meetings is really key.
Am I allowed to talk, please?
Yeah, of course.
Thanks, Prof, for your presentation so far. I just have a quick question on your biography. I think you stated that you have developed and regularly run a course to teach phage biology to African academics. Can you just tell us which African academic institution you probably
sure.
that have
So
this program?
Yeah, of course. So this was a collaboration between myself and somebody called Toby Nagel, who runs a non-profit organization, which what we would call in England a charity, essentially to empower academics throughout the world, particularly in developing countries, how to use bacteriophages. So she had this idea because she'd spent much of her life developing drugs for things like HIV, and she was fully aware that very often actually countries that need drugs are not the best first to get them. So she had the idea that instead of phage technology just being developed in Europe and Africa, in Europe and America, it should actually be developed in countries that really need it. So we could work with African people who are already academics, so vets and medics and food microbiologists, to show them the technology and the power and teach them how to work with phages. So she had this idea, but she needed an academic to help her develop the academic content. So we've now given the course 4 years, well, 4 times in Africa, once in Malaysia. The first time we did it in the University of Kampala, And then we did it that's in Uganda. And then the second time, but we had students from many East African countries for that one. And then we had it in Pwani, so in the south of Kenya. And again, we had mainly Kenyan, Tanzanian, Ugandans. We also had some lovely Rwandan scientists came with us. And then we did the course in Ghana. And then we had there were some lovely Nigerian delegates also attended on that course. And then the fourth time we did the course in Tanzania. And we've run it one more time in the university in Malaysia as well, but unfortunately because then COVID happened. So we did the Tanzanian course just before lockdown. That was in that year, the end of 2020, just at the very, very beginning of 2020.
Yeah. Clokie, I would advise if you can continue with your presentation, and for now we want to ensure that we address the questions after you are done, so
Okay.
it's of more ease than having a break in the presentation. Thank you.
Okay, and that's no problem. I shall just now move to talk about the other opportunities for scientific engagement, and then I'll just conclude the talk after that section. But I have quite a few slides in this area because I think it's really important when it comes to engagement to talk to wider audiences. And the reason for this is I think it can help take your talk, take your work from being sort of purely academic to potentially developing products, and also it can really help to promote your subject to the next generation, and can also link to funding, and in general it improves your communication as well. So this picture here is an example of a type of quite a very nice forum, which is a conference in Egypt, where there'll be 4,000 young scientists, largely between the age of 16 and 20, who come to this meeting to understand what the bigger issues are. So it's called BioVision. So I've had the opportunity to present there a few times, which has been wonderful. So the currency in a way of being a scientist is that you get is you have to produce papers and grants. And in order to get those grants, in a way, by communicating more widely, you get your work known, which can also help. And it can help you get your work, as I said, translated into products and also dissipate worries. So here we can see a picture of a friend of mine who also teaches on the African workshops, called Ben Chan. He's from the University of Yale. This is him treating a patient with phages. And he spends a lot of time communicating those aspects of his work to television shows, radio, and so on. So sometimes you don't know where your opportunities are going to come from, so I think it's important to be quite open-minded. So this was a nice opportunity I had. I was just on a train coming back from London, and I met this man here called Ben Jackson, who just happens to be a radio DJ. So I was chatting with him just about life in general, and then he asked me if I would be on his radio show, and I did. And I think it's important, actually, as a young scientist, you often don't think that you're as interesting as you are. So we all have stories. And sometimes if people ask you to share them, it's important to say yes. I also think it's important to talk to not just scientists who care about your work, but actually those communities. So here is an example. I've been working on a project on Lyme disease. So Lyme disease, you probably know, is a disease that's caused by a spirochete bacteria, and it's carried by ticks. Now, these ticks bite us and they give us the bacteria. So first of all, I was mainly communicating this work to academics, but I got a lot more success after I started communicating it to people who had Lyme disease and people who worked in communities that Lyme disease is very common. So this is us presenting our data at the British Shooting Society. So people that spend a lot of time outside doing activities like shooting, they have much higher rates of Lyme disease, so they're very interested. This is a problem. They were able to help us by providing samples and other forms of data. So can you see here as well, this is really important. So when you're communicating with a non-scientific audience, you have to be super clear and be able to present your work in just a few sentences. Other audiences I think it's really good to engage with are things like politicians. So this is in the UK, for example, we have a competition every year where you can try to get your science included into something where we show all our politicians about science. And Other people I put here are things like grant funders. It's important that the funders know what you're trying to do. Other groups might be patients. And then, for example, if you're working on animals, it's important to communicate with farmers. And also to the general public, including young people such as children. So doing outreach events at schools can be really enjoyable and useful, and you can get your work out there. Other types of audiences that I've enjoyed communicating with have been, for example, things like theatres. This was a really good, actually. I've spoken a few times to teachers. Teachers generally have to do these training events where they continue their own education to teach children. But that's very, very time efficient. I spent several times, I spent a day with about 100 high school teachers. So that was a really good audience in terms of getting the knowledge out there in an efficient way to younger people. I've done outreach events at schools with medical students and with scientific audiences, but in a slightly different field. So general, for example, in terms of other people that are interested in antimicrobial aspects. So this quote on the right, this is when I was in the theatre, and this is just the the theatre summary of my presentation. Okay. And sometimes it is really good to speak to students that you where you don't have an exact connection with, but they should be interested. So obviously I'm interested in phages for human therapy. So I was once invited to go and speak when I was a relatively young researcher to many 500 Portuguese medical students. And this is my diary at the time. I said that I returned to the UK quite positive about life, more big picture than sometimes, and a general feeling that we can all, if we want, we can engage with life a bit better to improve things at many levels. So I think often it's incredibly useful for you to speak to different audiences to help you contextualize what you want to do and how you want to do it. Other ways to engage I've listed here are things like advising museums. I've been asked to advise our science museum in London on how to communicate phages to the general public. So again, a very efficient way of communicating our work. I've talked at schools. I've also produced educational material for young students. Okay, how do you engage in general? I mean, there are other, obviously, to do the social media. I do a little bit on Twitter. I'm not actually very good on Twitter. I don't really tweet that regularly, but I do have quite a lot of followers. And like sometimes if I ask a question, and I'm quite interested to see the responses that I get, and I do find it a useful way to connect with people and to find specific phage papers. There's also obviously various small videos. It can be really useful to make little videos about what you're doing. Several videos on YouTube on different aspects of phage work that I've done. So however you like to interact with social media, at least that's one way that you can reach a lot of people. Now, when it comes to communicating with a wider press, you've got to be really careful that you don't use very complicated words. So, for example, I learned this very often at an early age. My father worked on seaweed, and when he did a lot of TV work, he was not allowed to use the word algae, which used to frustrate him. And then I think it's also important to not say yes to everyone who wants to talk to you. So in the times of COVID, I was asked to talk to a lot of people about the coronaviruses, and I actually said no to those invitations because I didn't want to talk about viruses that I didn't know as much about. So in a way, you define what your subject area is and what you want to talk about. And I'll generally speak to good, what I call mainstream media. I try not to talk to the very sensational press, which we have a lot of here. I'm sure you do as well. And then you've also got to be quite careful. So I've said no to things. For example, I was asked to be in a TV show that was looking to see, so I wanted to use bacteriophages to look at the transmission of viruses between people when they were kissing. So I didn't want any sort of bad associations with phages, so I said no to that. Okay, so one of the biggest audiences and the things I've done that I think has a lot of reach is to be involved in high quality radio and podcasts. These have amazingly big audiences. They'll reach hundreds of millions of people. And sometimes if you have a good producer, it's quite easy to do these things, but sometimes you have a a journalist that really doesn't know your subject at all, so you almost have to interview yourself. So it's really useful to try to think about this in advance and be super, super clear about what your story is and what you're trying to say. And then when it comes to radio, they often it's nice to sort of think about things that sound nice. That makes a big difference when you're trying to communicate. So here's some, I've got all the links here. I can circulate these links separately if you want to hear any of them, but these are the things that I've done along the way that's had a massive difference to my research. So I've talked to different BBC radio shows, both comedy shows, shows that focus on scientists, on shows that focus on health, and in general news programs. So these are different links. Naked Scientist is a very nice radio show run by young scientists. And I've been on more specific radio shows as well. For example, there was one on a radio show just for Clostridium difficile called Spores and More. The example of a little video that I made is given here with Microbiology Society. That describes their Tanzanian work, if anybody wants to watch that. And this is me explaining science to what we're doing in our lab, trying to use phages to the main BBC Science correspondent called Palab Ghosh. And this was a program we were at Jamal Khalili, who has a program where every week he interviews a scientist in the UK. And this is brilliant, actually, this program. I've listed actually here of all the different things that came from being on that one radio show. So in a way, the best thing was I was approached by a charity who now fund our work on urinary tract infections. And I have a collaboration with a major biotech company who heard me on this radio, the head of global sales. I went to a school to meet the granddaughter of somebody who discovered phages. I was approached by different people in different areas of science that I could then talk with, people I really would not have reached in another way. So this was also written up in a book called The Virus Hunter. And so I was able to reach some really good audiences, and from that a lot more media opportunities came along my way. And so I put here so this is one thing that came from it, Thermo Fisher. So it was the head of the sales for Thermo Fisher had this radio show, and that led to a very interesting collaboration. And also, actually, I should have said actually in the last one, I also wrote an article in their Thermo Fisher's little journal that they have for their employees. So that when it came to, this was arguably, as I said, one of the best things that came from being on that show was I was approached by a charity to work who enjoyed what I was saying about urinary tract infections. So they said, well, what do you need? So I showed them what we needed. They came and visited, and that's led to the funding of a really fairly major project that I'm leading now on urinary tract infections. So I just wanted to end my presentation by saying that those skills that you develop in terms of your engagement and working with other people, it doesn't just stop. So in my Phage Centre that I'm just setting up, I don't really need to see all of the details on this, but essentially I wanted to show you that so I'll be running this Phage Centre at Leicester. The idea of that's going to be to do a lot of phage work to underpin the development of phages for therapeutic use. But to do that, I will have to collaborate a lot with other centres, so microbiology centres, a precision medicine centre, the School of Chemical Biology, Maths and Advanced Studies. So in a way, I just wanted to say that the things that you learn to do with engaging with people, it's really, really key, not just to the early aspects of your career, but later on as you go through. So my final thoughts, so I want to leave a bit of room at the end of this session for a bit of a discussion, are that I think it's really important that you try to, within your work, find something that you find generally interesting, and then talking with lots of people can allow you to really shape up your ideas. I think it's really important to follow up conversations. You never know where the engagement leads. And then you can, it's nice to go from communicating to trying to get some preliminary data to formalize those collaborations. So with that, I shall return to being able to see you. I'm going to stop presenting.
Thank you very much. Professor Clokie. Okay, so you're done with the presentation. Thank you very much, Professor Clokie. You can clearly hear me, right?
Yes, I can hear you. Thank
Yeah,
you.
so that's a very brilliant presentation from you. We already have questions to address, and before I go ahead to read them, I would like to mention that from the details of your presentation, I can see a huge chunk of science communication going on there. From all what you shared across medical students across different tiers of the community, the global community, talking about politics, talking about not something just not targeted at academics even. So
Yeah.
We had a lot of sightseeing to see that yes, this is not something just focused on one aspect. It was like you ensured that your early career research reach was across a lot of sectors, let me put so, and that's very brilliant and very motivating. I would also mention that I think in our last webinar we also had a discussion that was focused on science communication, and that was Jessica Sacha from Phage Directory. She was
Okay.
giving the talk about that, and I see the similarity of how heavy
Yeah.
you are involved in that also. That's very great. So for everyone here, if you have questions, you can keep dropping them while I read out the questions we have already. So Professor Clokie would help address that. So we have a question here from Anzada. It reads, thank you for the insights, for the useful insights, and for taking time from your conference. From your point of view, what is it that we should look for when choosing our mentors, and what should we expect from them? So vice versa, what should we look for
Yeah.
when choosing mentors, and what should we expect from them?
Yeah, that's a really good question. And actually, at Leicester, we run a mentorship scheme, and there's almost like a contract so that the expectations are known either way. I think what I was referring to, in a way, it's a little bit less formal than that. So when I was a young scientist, I looked for people whose work that I really respected, and I wanted to work in a similar area. And I would often just ask them questions about their work, but also just about, well, how do how do you go about this? How do you go about writing a paper? How do I go about trying to attract funding to this area? So in a way, it's people whose work that you like, and sometimes it can just be perhaps people that you like that maybe are in a slightly different area but are successful at doing what they do. So I would look for people that you respect and a little aspire to be a little bit like them. And then now I've noticed that I now have a few people. So some people I mentor through our university mentorship scheme. So I have the things that I think I'm good at. I'm quite good at writing grants. I'm quite good at writing papers. I'm quite good at chatting, talking. So the people want help in those types of skills. They can come to me. But then I just have a few young scientists throughout the world who a little bit like how I did with other people. It's now my turn. I'm a older scientist now. It's my turn to some extent to give back a bit. I mean, I still have mentors. I think you need mentors at every stage of your career, but now I had so I think you look for people who a little bit that you would like to sort of a little bit follow in their footsteps, perhaps. And I think you can't expect so much because people are really busy. So if someone says to me, I don't know what to do with my life, I don't really know how to help them. But if they say, I've got this idea, I don't know how to develop it, or if I should be aiming for this or that, then so I think in a way your expectations need to be not sort of, you need to be quite proactive, but just where you need a little bit of some steering to help you get to the next stage.
Yes, that's I very much agree with your answer. Actually, the emphasis on the contribution of mentors, and I think that is also one stronghold that we run with back here in the team of Ibadan Bacteriophage research team. We also have faculty mentors that they definitely guide us in the things we do. So we reach out to them with advice rather, and for suggestions that we always welcome. And I'm also glad to reiterate and mention that some of our faculty mentors are here. Once again, I would like to recognize the presence of Professor Ademowo here with us. Is the overall faculty mentor for the team, and then we also have Dr. Adewumi. If you'd like to speak, please, sir, you can unmute to speak to. And while we wait for that, there is another question from Williams.
Hello.
Okay, yes, you can go ahead, sir, Dr. Adewumi.
Yeah, good afternoon for those of us around there, and good morning out there. It's been a wonderful time, and I quite appreciate everyone for your participation. Listen to Professor Clokie. It was a great one, and for our guys here, they are doing beautifully well, and we are really proud of them. So I'll be glad to, though it's going to be in and out, I may not be here all through, but I'm really happy that we've come this far. Thank you very much. I'm certain Professor Ademowo will be speaking too. I can see he's on. Thank you very much.
Thank you.
We hope to reach out to everyone over time.
Yeah, that's fine. Before we proceed to the next question, Professor Ademowo, I would like that you you unmute to address just for about a minute, if that will be fine, sir. Okay, it's is on. Sir, you are currently not audible, sir.
Hello, but it's on already. Oh, I guess it's probably okay. It's not in the office. Maybe
Yes, sir. I try unmuting so we can hear you, sir.
I guess is at the car park.
Yes.
maybe just move on when he gets to the office.
Okay. Yes. So if he's able to join, if he's able to talk to us later, that'll be fine. From Williams, I would just read that now. It says, thank you very much, Professor Clokie. For regions that don't have established media platform, what recommendation can you give to help them put their voice out there?
Yeah, that's a very good question. I would say you can definitely engage with things like, for example, through social media, things like you're making a lovely job of advertising the network that you're establishing within Nigeria. So I think through things like Twitter, other open platforms, and also, for example, I'm the editor-in-chief for a journal called Phage, Therapy Applications and Research. So you can write, you can write some more general articles and things like this. So it's a question of, or perhaps even being able to work with other outlets. So even though you're based in Nigeria, that doesn't mean that the BBC isn't going to be interested in you when it comes to the sort of specific, more specific things that are of global concern. So I think it's a mixture probably of looking at open platforms and then also seeing where you might be able to contribute to other platforms from other countries. And I've quite often been involved in media from countries, not just the UK, so from America, Canada, different European countries, even Russia. So yeah.
Right. So we have one more question, but before then, can Professor Ademowo try to unmute now to speak? I noticed you were able to do that while Professor Clokie was talking.
Hello?
Yes, you're audible now.
Okay.
We can hear you, sir.
Thank you. Yeah, I just want to appreciate the the speaker for a beautiful lecture. I mean, all of us have learned a lot from the lectures, and I want to believe that we will continue to collaborate with her and others, and I believe strongly that whatever we have been able to learn today, new knowledge that has been acquired, I mean, we will try as much as possible to ensure that it is disseminated, and we'll make a lot of gains from it. The IBRT of the University of Ibadan is a group of undergraduate students who have tried as much as possible to put together this kind of lecture annually, and also seek some form of collaboration from people around, people who have different knowledge. So I just want to appreciate you all, and also to actually also appreciate our students who themselves are also participating in this. So thank you very much.
Thank you very much, Professor Ademowo. Thank you very much for that. So, Professor Clokie, we have one more question, which is a very fantastic one. It's from Wambuchuku. Uzumbechuku. I'm very sorry with that pronunciation, but the question reads, "Hello, Professor Clokie. Thank you for your presentation. Quite inspiring. Now the question poses that what will be your specific advice for a first-year PhD student just starting a bacteriophage research?" I would say that this is one of the motivations to setting up your session. So kindly approach that.
Yeah, that's a brilliant question. And I was actually just speaking before I came to Ireland to a new student of mine who's just starting his PhD, and I was telling him that I think key is really treating your PhD like it's also almost like a your own business. Like the more you put into it, the more you'll get out of it. So first of all, you have to first of all, spend some time thinking about what your area of focus is going to be, discuss it with your supervisor. And then it's very easy to spend a lot of time just thinking and reading, but actually you need to start to generate some data, and then that will help with your thoughts. So I think when you're starting your PhD, it's a mixture of really thinking about what it is you want to find out, and that to begin with, a lot of the things that you will do are probably chosen by your supervisor. Your supervisor most likely will have some idea of the direction of something that they want to find out. For example, can we find better phages to treat mycobacterium tuberculosis, or they'll have a question. And to begin with, that supervisor will direct you. And then there's this lovely sort of crossover at some point in your PhD journey, where probably on the basis of your research, you'll say to your supervisor, "Oh, I really want to do this, and I'd love to investigate that." So you have this lovely sort of journey between the supervisors and the students, where in a good relationship, it should be it should be the student having ideas but being gently steered, so you don't waste a lot of time doing experiments that perhaps have been done before or are not likely to generate data. So I'd say work hard, talk to your seniors, talk to other people that do similar research, and treat it like a proper job. Be in the lab nice and early every morning, and work nice sort of full days, but not sort of crazy hours, but just nice full days. Treat it like a proper serious job, I would say.
All right. Yeah, that's a very fine answer to the question. Well, we do have one hand raised and one more question, and we want to try as much as possible to have this addressed in about 2-3 minutes. So I would read the last question we have and then give Newton the talking space to quickly address his question also. So for the question, it says, "Hi, Martha, what's your advice to early research career in specifics, postdoc fellows trying to get established in their work, on their work? So what's your advice for postdoc fellows trying to get established?"
Yeah,
Joseph.
I think Yeah, I think that when you're doing your postdoc, that's really, really key, isn't it? Because at that point, I think you want to get good at doing one thing, so you build up a name for yourself in one particular area. So people will come to you, think, oh yes, Joseph's brilliant at doing those experiments. I must involve him in my future work. So I think it's really good to really focus at that stage on producing data. You really need to produce data and get known for that one thing. So I think it's really key during your early postdoc to take up you try and go to places where you'll meet the next people that you want to work with as well. So if you're working on particular phages and you know that you really want to work in that area, I'd say don't feel shy as a young postdoc to write to people and say, I really like what you're doing. Is there any chance for me to come and join you, or could I get some funding even to visit you and work with you? So I'd say get good at one thing and then reach out to other people. You can be very proactive at this stage, and you're probably much more desirable. I think often you can think you're quite alone and you feel like you're bothering people, but never feel that.
Yes, I would also like to add that's like a sort of advice that extend across reach undergraduates, PhD.
Yeah.
So try to reach out, because we also plan to apply that myself and everyone here. So it's not like specific to postdoc fellows alone. Definitely reach out, express yourself. So Newton, please, in a minute, can you quickly speak and
yes, thanks. I'm gonna be very quick. I just want to encourage I mean, student working on phage research, or doing carrying out phage research in Africa. The undergraduate student, the postdoc, or the doctoral student that I'm actually fully graduated PhD student. I completed my PhD on phage research here in Africa, and I can tell you that it is not an easy job. You need really to be engaged, to be committed, and to always be online to collaborate with the other people doing, the researcher carrying out the phage research in the Western countries. I would like really to take this platform to thank Dr. Jessica, and also she has really helped in that journey. Also, that thank Dr. Atunga at that's with the Kenya Institute of Primate Research. We really work together. You need collaboration to achieve your goal, to achieve your objective. Thank you so much.
Oh, thank you, Newton. Anza, do you have a question?
Hi, Professor Martha. Can you hear me?
Hello. Yes, I can hear you fine, thank you.
Thank you so much for the great presentation. I've been following you on Twitter, and I really enjoyed your tweets, your papers, and especially congratulations for for establishing the Phage UK Therapy Center. And we in Pakistan are also working on phage products, and we have developed a phage cocktail against poultry colibacillosis. So we as a student group also presenting this idea at different platforms and platforms national and international. So we would love if you can also arrange a session with us, because we have a lot of enthusiastic students in Pakistan as well, and they would love to join. If you can, if we can write you through email.
Yeah, you're most welcome. I'd be very happy to do that. Thank you.
Thank you so much, ma'am. And ma'am, one more thing I wanted to ask. Can you please, we are working on phages. So at this stage, before my PhD, I'm really enthusiastic to learn different things about phages, like in vitro testing, then whole genome analysis. Can you please elaborate a little bit, like on which particular skills we should focus to get into really a good school, or to get, or to contact a supervisor who will be willing to accept us after having those skills? Thank you.
Yeah, I mean, it very, sorry.
Yeah, no, I want to inquire if this would be like an elaborate if it's going to be like an elaborate, we can elaborate answer to it, like based on a question, we can have it set up like an email instead if it's going to take time. But if it's going to be like a succinct response, definitely, Professor Clokie, you can go ahead.
Okay, I'll just answer very briefly that I think that what people will look for in a student is good thoughts, hard work, and tenacity. And in some ways, if somebody already has, if somebody doesn't have particular skill sets, I can teach them that. But you can't teach sort of motivation and enthusiasm and hard work. So in some ways, I wouldn't get too occupied by learning a specific skill before you go somewhere. But you're absolutely right. You've identified you do need for to be a phage biologist, depending on which bit you work on, there are different skill sets that it's very, very useful to develop, both bioinformatically and phenotypic characterization. So and the more skills you have, the more attractive you will probably be to getting independent positions and further postdoctoral opportunities.
Yes. So, Janet, you are last. You can have the floor to speak now. So we will be taking your question as the final one for this session. Yes, go
Yeah,
ahead.
thank you. Yeah, thank you so much for that opportunity. And it's really nice to see Martha giving you all these tips. And I want to say that you're really fortunate to have all these tips at this stage of your lives, because some of us have to learn and follow the trend, and learn on, with our mentors, but at the same time trying to pave way for ourselves. So it's a golden opportunity. All that you have heard, take it and learn it and put it into practice. And I have worked with Professor Clokie for over 10 years.
Wow.
Actually, 13 or 14 years, if you add my PhD and my MRes time in Leicester. So I just want to add that I've got 4 things to say. One is have a dream. If you don't have a dream, whether you meet the best person in the world, the president of wherever, if you don't have, you will fall out eventually. There are challenges, there are problems, you have to stand firm. The only way you stand firm is through your dreams. Your dreams are like your pillars you hold on to, no matter what you go through, you hold on to that dream and keep going. I have had several challenges, but I'm telling you, I had a goal, I had a dream, and with the right people around me, I was able to pursue this dream to the very end. I am from Nigeria, from Kaduna State. I had my first and second degree from Nigeria, Ahmadu Bello University, and I'm telling you where I am here is because of the dreams I have dreamt while I was in Nigeria. Secondly, your dreams might not be straightforward. For some people, they have very straightforward dreams and their goals come to pass as they have planned it. Sometimes you will have plans. Your plans will not come in the way you think. So you have to take opportunities, you have to take small steps, you have to be challenged, you have to make sacrifices. So if your path is not straightforward, don't be discouraged. Keep pursuing it with the right dreams, with the right motivation, and the right people around you. You will make it. Then take small steps. I know some of you are motivated now and you might want to go onto the telly, you might want to start writing big papers. It doesn't come that way. You have to start small. You have to go through, small publications. You build your platform till you reach this particular position. Professor Clokie has been over many decades doing this work and reaching this position. I am still learning as well. I have got people to look up to. So don't feel your dreams will have to be fulfilled the next day. You have to follow the path because you have to learn the process. It's not just the outcome, but the process. The process is what builds you. Lastly, be humble. Okay, I've had people I've worked with as a postdoc in Professor Clokie's group. People are not really ready to learn. You have to be humble. You have to know there are people that have gone ahead of you and you learn through them. So I hope you will take this opportunity you have got, to build your platform in your area, starting with your dream. Thank you so much. And nice to see you, Martha.
Thank you very much.
Thank you, Jen. Yeah, I think that's a perfect words to end the session. It's wonderful, Jan. I mean, I should say though that not only did Janet dream, she was just incredibly true to herself. You know,
Right.
she it was not, life is not always easy, as she says, and Janet took every opportunity, and I couldn't be more happy to have worked with her, or proud of where she's got to. So thank you, Janet.
Thank you very much, Janet. I think we are definitely holding on to those pillars that you mentioned. Definitely, I can assure you. And all undergraduates, all graduate students, and all audiences here are going to key into that. It's a very nice way to cap this session, really. So thank you very much, Professor Clokie. And thank you very much to all those that are present for this session. So we would be having another session coming up in the next 15 minutes, and that is by Dr. Temitope Faleye, and that is going to be another brilliant session of this particular webinar series. So we say thank you very much to all that are present, and do join us in the next 49 minutes to have us discuss another session on this World Phage Week celebration webinar series. Thank you very much all.
Thank you so much for the invitation. It's lovely to chat with you. I wish you all the best. Bye bye.
You too. Bye.
Bye.